That's what I usually do. Okay. Will,
Sure.
you want to go first?
Yeah,
Okay.
yeah, hooked up and everything.
Oh, I'm sorry, I didn't see you slip in. Would you like to do an introduction, Dr.
I
McCullough?
think we'll well known to everyone.
Thank you. Is there any,
Still do whoever's lab is bioengineering.
thank you. Is there any way to keep track of time? I will get lost.
Right there on the door, on the right
thank
side of the door.
Thank you very much. I
Mhm.
will otherwise get lost on tangents. So I am going to talk today about some work we recently just submitted to a journal called LCSS Control Journal. You're going to be familiar with it. It's on what Sylvia actually spoke about last time and extended to some degree. So generally our work and most of Sylvia's lab works on this topic of reachability, which is given a nonlinear or linear system, some sort of system, you want to compute some way of optimally controlling it so that you're robust towards stochastic disturbances or worst case disturbances. But again, if you're robust towards a worst case disturbance, you're robust towards a stochastic disturbance. And my interest as a bioengineer particularly is extending these models to higher dimensional settings. So these like control algorithms are really great and they have some very beautiful theory underneath them. But in general, when you try to actually compute the solution, you need to do some sort of solving over a grid, like to solve a PDE, basically, that I'll talk about in a second. And these methods, therefore, grow exponentially in compute with the size of your grid, i.e. the dimension of your system. And so they're limited to small dimension systems, which in a lot of cases are toys or reduced models. And as most biologists will tell you, that's not very applicable to the many metabolic, multi-scale, or finite element models or anything that we come across. So that is the general motivation, although I'll start on more simple grounds today. So again, this idea of a reachable set, before I get into the rigorous justification, is that if you have some region which is a target, but maybe not a target you want to reach, but also a target you want to avoid, you want to know, given a system, which states are going to hit the target in some sort of allotted time window, because maybe you want to reach it or maybe you want to avoid it. And again, this is under some sort of stochastic disturbance. So in the reachable sense, we ask for a set where there exists some sort of control trajectory, some sort of way I can operate my system to get it to the target despite a worst case disturbance. And in the opposite setting, we would talk about despite any control trajectory I choose, there is a disturbance which could take me to this target, and therefore I could be unsafe. So again, my hope is to apply these to higher dimensional models. To name a few, I'm motivated by like finite element models where you could do some sort of radial basis interpolation, as is done on the meshless models, to compute inner stresses and strains. However, these end up being very large pseudo-linear models. And again, with the dimensions we can approach or we can tackle today with Sylvia's existing algorithms, it's like 6 to 10 or something. And in general, Amazon funds Sylvia and most of and some other research groups in Franklin Antonio that do general like drone and supply chain robotics. So you often want to see these if you're operating not just the specific dynamics of some sort of robot, but also many agents at once. This can quickly become high dimensional. And when I see these problems, I think about lots of cells and how you might drive a population of cells particularly, but that's outside of today. So again, we have a system, which I'll call F basically, and this is a field which has some, and X dot is this notation to describe the dynamics. So again, we've defined our system such that it has some sort of dynamics and there's some sort of control U and control D or disturbance D, and they're playing against each other. So here is a two-dimensional space I have on the right with some sort of field F, some sort of nonlinear field. You can think of it like a wind or a fluid, or more abstractly, like the forces on a pendulum or something. And we have some sort of initial condition in our space, which again could be state, velocity, acceleration, and if you were to take this from a literal physics example, or it could be something like concentrations of a metabolite. So generally, what we're concerned with is ascribing some sort of cost to our operation. So the point is we can maneuver around these spaces, but we want to do so such that we're optimizing some sort of function. And this can be abstract in general. And so let's first just talk about a cost function, which has two terms, one which is a function j of the final point, x capital T, and it also has an integral for the running time. So you have a running cost as well. So you might think of these as one, a final cost could be something like where ultimately did I end up in relation to a target? So that the metric would be a distance, or you could also have a running cost like energy consumption, for example. Again, we have multiple trajectories here that might go to different places, and we particularly will have some sort of target set capital T calligraphic here, which is in the upper right hand corner. And you pick this function j and the running cost L, particularly so in the business of safety. You want this property such that your cost function being less than or equal to 0 for some sort of control and disturbance means that your final point is within a target. And again, that generally means that we don't really care about the U and D that occur along the pathway. We're more so just concerned with getting or not getting to the target. And we can consider the optimal value of this cost for some sort of payoff that we have previously defined, such that this is now the optimal cost, given that you have some sort of control trajectory and disturbance trajectory. The int and the soup here are just mathematical notations for minimum and maximum, but are needed because the min or max may not exist. But in most cases that we would talk about, they do exist, and you can think of this as you're trying to find the trajectory U, which minimizes the maximum disturbance or the maximum cost that the disturbance can get to. So again, this would be like if you're trying to avoid this target T, you might want to maximize the negative distance or something. So again, that would be your disturbances goal, and it's trying to get you closer to this target, which may mean that your metabolic profile is of some sort of concentration, or you have some sort of state and velocity again. Particularly though, the reason we care about this optimal cost is because it satisfies this property such that now if your optimal cost is less than or equal to 0, it means X is in this set R. And this R is a reachable set. And that means in the reach case, there exists a U trajectory which can drive us to the target in some final time, such that no matter the disturbance that occurs. And it's the same math for reaching or avoiding. But let's just talk about the reach today, basically. So there was this famous theorem in the 80s when this work was sort of pioneered that showed that if you want to solve this optimal game, this differential game between your control and your disturbance, and you want to figure out where each of them wins and at what time horizon, it's equivalent to solving this partial differential equation here, which has an evolution on time. So the value function evolves over time with relation to the negative growth of your Hamiltonian, where the Hamiltonian is defined on this bottom line here. And again, it's this, now it exists exactly, and can be considered over the minimum and the maximum of something called an argument called P dot producted with our original system F. And that P is often called the costate as an aside, because P here actually represents the Lagrange multiplier, if you're familiar with Lagrangians. If you were going to take the Lagrangian of this cost function here, P, you would have to ascribe this constraint, which is the system dynamics, and that's where you include P. And that's what leads to this form in the proof. So, and from this, we can actually, after solving the PDE over space and time, we can come back and control the optimal control or the optimal disturbance as well afterwards. And that's how we operate controllers, basically, from these. Again, the problem here is that this is extremely burdensome. So with 2- and 3-dimensional systems, we can do this more quickly even than this plot, but not on the scale that fixes it. It's just more so that we can get it. You can get 3-dimensional systems down to minutes these days, except it can be up to an hour. And 4-dimensional systems are hours, and you have to come up with some very sophisticated methods to do higher dimensional systems, and they're still limited to systems which have specific forms, which can be sort of parallelized, if you think about it that way. So my goal here is actually to bypass this impossible bridge that I have nicely depicted for you all. By doing something which might seem a little abstract, but we're going to take this nonlinear system, which is high dimensional, and we're going to transform it into a linear system. And we want to do this in an intelligent way so that it's not localized, basically. And more recently, previous authors at UCLA have devised this hop solution for fast linear systems. And it's not a free lunch because it still involves solving a hard problem, but it's not necessarily sensitive to dimension. So we have some new gains in this pathway, which hadn't existed before. Again, this hop solution only applies to linear systems, as I'll show. So we have to come up with some sort of linearization method. And the problem is when we're solving this PDE over space and time, if you have a linearization method that is localized, you'll incorporate some sort of error in certain specific spatial regions. And that can lead to erroneous solutions that propagate through these erroneous regions. And therefore, you would get pretty bad solutions to your quote unquote viscosity solution. And so you want a balanced linearization. So just quick background on this hop solution, which I unfortunately won't be able to talk about in detail or the derivation for it. But at a high level, you can solve this value function V. Here, this is just a notational subscription so that you are solving it backwards in time. By solving this optimization problem, which particularly corresponds to the dual solution, if you're familiar with duality and optimization. For any optimization problem, there's a sister optimization problem, which you can solve in order to get the same optimum. And that is ultimately what the hop solution is doing. It's a bit complex. So again, we're solving a cost function now, an objective, which has first off this term, which is something called the convex conjugate. Don't worry about it if you don't know what that means. But we also have this integral of the Hamiltonian. So for any time point, we have a long integral, which we will numerically solve as a sum of many, many small time points. And you can imagine this cost function being complicated to solve. And particularly, this form is only valid when we have a linear solution because they recognized in 2016 that this linear solution can actually be transformed into this solution, this system here, which you might note actually lacks a drift. So it lacks a field. So this system here had some sort of AX term, and you can remove this by actually mapping it with the exponent of the negative TA, which is the solution of the linear system, if you remember your ODE class, to this state space where you don't have a drift. It's just control and disturbance, but there is a time varying map to get here. And for this system, particularly for linear systems, which can be mapped like this, which is all linear systems, you have this nice explicit solution. So they showed in their work that it's amenable to high dimension. On the left, you have computation time per point. And on the right, they're plotting some BRSs, some backwards reachable sets. Again, this is computation time per point because the nice thing about this explicit solution is that we now do not need to propagate a PDE over space and time, right? This is one thing you always learn about when you're solving PDEs. It's the trouble with solving PDEs numerically is if you have to propagate over space, that means that at any time point, you need to take some sort of numerical approximation in a neighborhood of your point of interest. So that generally means checking some points in each grid dimension. And that's exactly where the compute will grow exponentially. So this means that we don't need to propagate over space and time. Again, we can solve each of these points independently in the value function. So this was applied by some of our now collaborators at the Naval Center in China Lake, particularly to do multi-agent control. So now you want to optimally pursue and evade, they call these games, but you might see what they're used for, particularly for driving multiple agents towards one agent. So I asked them if I could use their code, and they gave me their code, and it was a mess, frankly. But it was also not intended for public use. And so at that point, I went back and forth with one of the postdocs at UCLA, who's now our collaborator and professor at UCR, Riverside. And we developed this package in Julia, because it's a fast language. And this is me just demonstrating that it produces the same plots for the same systems. And it's fast. It's actually faster in their implementation. Again, they were mathematicians who wrote this not for the sake of optimized speed, but just to show that this method would be much faster anyway. And so this could also, this green line is our most recent package speeds, and it could also be improved. By the way, if any of you have any questions along the way, please feel free to interrupt. So again, the beauty of the hop solution is we get an explicit solution, do not need to propagate over space and time. So they're decoupled. The trouble is, first off, this only works for linear systems. And what I'm not going to be able to talk about today is even for linear systems, solving this optimization problem can be difficult. And therefore, there's a lot of further work to be done, but we are excited to take it on. So now I'm going to talk about the other side. So this actually says ML Koopman, although I am these days, and I'm only going to talk about deterministic methods of linearization of this global linearization today. And hopefully we'll talk about some neural net approaches in the future. But again, we're trying to now linearize the dynamics so that we could start applying some sort of high dimensional multi-scale models, particularly to this workflow and show some sort of safe or robust operation of them, where otherwise some sort of minimal controller failed. So it's based, this linearization method is called Koopman theory, which is jokingly called Koopmania these days, often because it's got such a cult following, but is definitely over applied without the rigorous justification. And it dates back to this paper in the 1930s with Koopman, Bertram Koopman, I think, and John von Neumann, who showed that any nonlinear system that I described before can be mapped to an infinite linear space. So in this infinite linear space, we have a perfect representation of our nonlinear system, and that's where we want to do control. The problem is that this proof doesn't talk about A, what happens when you approximate it with a large matrix, which we obviously have to do in practice, and B, how to get to this space. So the point is they have some function G, basically, of our original system, but they don't describe how that G should be formed. Should it be a polynomial? Should it be a basis of, maybe you're familiar with random Fourier features, which are just basically Fourier terms. And you could also choose some sort of radial basis function. These are all popular, but for none of these was the proof actually used. So that is left to us in deciding which works best. And also there was no mention of control. So we are often using this form here at the bottom, which says we're trying to find a linear system, which again is some G mapping of our original states, which also includes some sort of linear injection of control disturbance. So maybe you can better understand this with the quote unquote textbook Koopman system. So if you consider this nonlinear system on the right, which is two-dimensional and has these simple terms for its derivatives for each dimension, you might notice that if you define a change of bases such that you have a third variable G3, which is equal to x1 squared, the change over time of this third variable will be 2x1 times the relation of x1 with respect to time. And so it becomes itself and therefore can be exactly linearized given by this matrix on the right. But in general, most systems cannot be exactly linearized finitely. So this is just demonstrating that our package works. So again, this is us computing the value function with some sort of PDE solver here on the right, just for a sanity check. And you can see that this exact system is perfectly representative. I shouldn't say perfect, it could be rigorously compared, but we again don't have analytical solutions for these. So you still need to compare two numerical approximations ultimately. Thank you. So we have some challenges in this. Particularly, we would like to preserve convexity, which I can't really talk about today. But we also need to define how our target changes in the lifted space. And we need to linearize, as I was saying before, with respect to a diversity over space. So this is really important because if our PDE changes over space, if our linearization is sensitive towards certain spaces, then our solution will be as well. And it could allow this sort of erroneous propagation of the solution boundary. And again, we still need control, which is troubling. So we have encountered many issues in this, although we are trying to tackle them one by one. These are this is my future directions slide, basically. But Anyway, to give you an example, so this is the Duffing model, which models an elastic pendulum. And is just a common toy example in nonlinear analysis. And we can use Koopman to perfectly reproduce the trajectories or pretty perfectly. It's nice. And now here is our work. So the yellow solution is the value function when solved by lifting it to a 15-dimensional space and taking the lifted target. And the green solution is based off of a local Taylor series approximation. The black is our original target and the blue is the true system. So I want the blue and the yellow solution to match. And you can see that over time, the yellow solution is the best, although if we have time afterwards, I can talk about why it looks a little jagged in the bottom. This was our plot in the submission and the error compared. So the error between these two sets is worse for the green, the local approximation, significantly, particularly for long time horizons. And then I applied this to a 10-dimensional yeast glycolysis model. I particularly chose to add control to this system, which was autonomous, but I chose to control the external glucose concentration, the total ATP pool, so ATP and ADP, and the total NAD/NADH pool. And from this point, we then tried to linearize this high-dimensional model. And actually, it's simpler to linearize than you would have thought. So it's high-dimensional, but it generally has this oscillatory nature such that you can pretty well linearize it. You might see this orange line as the linearization for some short time horizon, at least up to 2 seconds. So we took this and we applied it and then used it as a controller. Again, I'm not plotting reachable sets here because they're 10-dimensional and hard to think about, but we can at least use this to generate an autonomous control. And so here I have one example of that from our statistical analysis or of just applying it many times. And you can see that this hop controller is able to drive the pink ATP line basically to a target concentration when 2 other popular controllers are unable to using this linearization. And this is because it ends up swinging the system, which is pretty interesting. So you might think, as I just tried to mess around in the first couple of days when I was exploring this model, that you would try to increase the total ATP pool, and that's a bad idea. That actually just generates a bunch of ADP. And the secret is to basically swing this total ATP pool, which is again the green line. And again, you can see this here, maybe just for clarification. These are the 3 states which I'm applying control to. And you can see this swinging that occurs by the controller. Again, these look very jagged because we have a random disturbance incorporated, but it works a lot better is the point. We have like a 96% success rate with this controller, while we only have like 20 in comparison controllers. And yeah, there are several directions we would like to take this, which I unfortunately can't talk about today. And this is the title of our submission, which is on the archive, and there's code associated with it on the page. Thank you so much for listening to me, particularly my lab and the 3 professors who helped this work a lot. Thank you. We have brief time for questions. Maybe I should do it at the end.
No, we have time for questions.
Yeah.
I have a real quick question. I just wanted to know about the dimensions. That part confused me, I guess, like, what does this word mean when you're using it? Because on that slide where you had like, the traditional computational methods, because it's exponentially high. I see that you're doing this 10 dimensions, so is that 10 variables?
Yes, that's 10 species, exactly. Yeah, so if we would like to apply this towards controlling a large cellular metabolic model, we would need something probably in the scale of like 80 dimensions, because there's like 80 species you'd be keeping track of. Maybe you could reduce this, I don't know, to 50 or something like this. But right now, the best I've been able to do is like 20. And this comes down to how well you can optimize that hop cost. So you need we applied some very simple optimization methods to start with, but now's the time to start bringing out bigger guns that people use to optimize higher dimensional cost functions.
Have you talked to Dr. Kallenbergs about a sort of a driving biomedical problem for your thesis?
No, not specifically. He has some high dimensional brain models they use, which I don't know of a specific application for this domain, but I at least know.
I think it should.
Okay.
Because, and this is a sort of a common experience of, I think, students in programs like bioengineering and chemistry and biochemistry and EMAE, where they're working on science problems, but their sort of training is more on the technologies that you bring to bear than on necessarily the scientific domain per se.
Yeah.
Is that it sort of goes without saying that technologies like this will be valuable in biomedical science because there are so many nonlinear problems and so many control related problems and so many safety related problems. So it's sort of easy to say, okay, well, if we develop this, it'll be useful, and in some sense it'll be true. But the thing is, when you get to the details, where you end up putting your effort and which way you end up going, and say how big of a system you want to be able to model, for example, or how important it is to be able to linearize, will come down to the specifics of the problem you're working on. And so that actually becomes your friend then.
Yeah.
Because then it helps you, out of all the things you could be trying to improve, it helps you focus on the ones that are going to get you to an answer. And so I don't think it's too soon for you to, even if it might require most of the remaining time just to develop the methods. I'd sort of encourage you to find that driving biomedical problem sooner rather than later. You know, they sometimes call the opposite strategy, build it and they'll come. And the problem is when you hear that in an NIH study section, it means we're not going to fund it. Because people have all had this experience of they build it and then people will come, but actually they don't come because people come because they see a similarity to their example to the one that someone else solved, and then that's how it goes. So I would recommend you know enough about what you're trying to do now, and you know enough about where the challenges are and what the limitations are and where the opportunities are, that you'll be able to choose a good problem. And you might have to go through, I always tell students too, like, you know, if you want to have good ideas, you have a lot of ideas. Actually, Einstein said that, right? So have as many ideas as possible and then sort of figure out the one that, you know, these approaches seem best suited to and could have the biggest impact on. But they could be anything from, yeah, fairly complex neural dynamics and control problems to cardiac arrhythmias, to glucose control, to immunology problems. There's a big world out there of problems.
Yeah.
But that's the problem.
Yeah.
They actually all have their own sort of constraints and issues and quirks.
Yeah.
Mathematically, they may have similarities, but the devil's in the details as to what you need to know and what matters. And
yeah.
That's why I think you're ready to at least start making a list.
I appreciate that. Thank you.
You're using vanilla DMD for the.
For the glycolysis, I was actually, and it's not very good, surprisingly. So the one I was showing before is a polynomial, and so EDMD here. This is EDMD. The plots, because they're nicer. The DMD plots are debatably ugly. It works. It gets you to the right concentration. The frequency is wrong generally, but the point is that's good enough. And we find again that this is just so I guess the point is that your linearization can still incorporate similar enough dynamics that control on it is still good enough.
Well, we should probably move on to Gaston and I guess you're going to give us an update on the book you gave in this.
Everybody help yourself and sandwiches, and before you leave, please make sure you sign the sign-in sheet, which I think is over there by Marcus.
Sorry, no problem. Thank you. Yeah.
Oh, I think Will's still sharing.
Oh, am I still?
Okay. Hi, everybody. My name is Justin. I am in the Varkey lab. I'm a second-year PhD student in the biomedical sciences program. And today I'm going to be talking to you a little bit about glycobiology. So we're going to take a little break from math. And by a little break, I mean a lot of break. And we're going to enter glycomania. I gave a talk at the Interfaces Symposium for 5 minutes, and I realized that I probably created more confusion and questions than I did answers. And that's because this is a really complicated field with a lot of history and a lot of intersection. So my goal today is to unpack that a little bit more, and with the available time, explain kind of where I'm at in the project and the methods. I don't want to go into too much of them because I really want to focus on kind of what has led up to this point and the novelty behind this. So without further ado, we're going to get started, and I just want to reiterate that, yeah, there are a lot of healthcare problems today, just by virtue of being in the Western world. We have Western diseases, and 2 of the most common are type 2 diabetes and atherosclerosis, and there's been an increasing incidence of cancer as well, of all different kinds. But there's this association with the diet and all of these conditions, and so an important question is to ask, what is it in the diet, or what is it in the diet composition that can be contributing to these susceptibilities and chronic disease development? You know, what are they, and what are the strongest associations between them? Well, it turns out that red meat seems to be kind of the glaring issue here. And that's why so many epidemiological studies suggest lowering rates of red meat, specifically processed red meat, to reduce the incidence of these conditions like cancer or heart disease or type 2 diabetes. But despite all these guidelines, it remains still one of the most consumed meat products in the United States, and we export this behavior to several countries, which also adopt those behaviors and land practices, which contributes to deforestation, more CO2 in the air, et cetera, et cetera, et cetera. So the Lancet says that a revolution of our food industry and the way that we consume is one of the biggest and most important things that we need to focus in on for the next century. So just very quickly, what are some of the numbers from one of the most recent studies before the pandemic? High processed meat was associated with 300,000 deaths, but worldwide it's about almost 900,000. Again, but there's no mechanism for this, okay? These are just associations. So what could potentially be going on here? What can't we see right now? Just a couple more numbers that I think are really important, because for my project, for my minor proposition, I focus specifically on colorectal cancer, but I just want to extend to you all that it's not just colorectal cancer. There could be other associations, but focusing just on this one specific diet-related Western pattern lifestyle disease, up to 50% of colorectal cancer patients do develop recurrence. And it appears that following a Western diet behavior, not a lot of fiber, higher meat consumption, higher sugar consumption is associated with recurrence and mortality. So in other words, if you develop colorectal cancer and you put it in remission, but you continue to follow a similar diet patterns, you're more likely to get it again. So again, what could be going on behind the scenes here? So for years, inflammation has been associated with chronic disease. It's driving it, promoting it, progressing it. We don't really know, but inflammation in general seems to be another underlying link between these 2 conditions and red meat consumption. So the whole cancer biology is very interesting in and of itself, and how cancer can manipulate inflammation to its benefit to sustain its ability to stay there. And it has been, a promising area for investigation and disease prevention. But again, what is initiating the inflammatory response in the body? So that's why I want to orient you to a nutrition label. And the nutrition label is, required of all foods that are processed and packaged, not any fruits and veggies that you can buy, obviously. But my question, even before I began my studies as a nutritionist, was that this can't be everything that's in food, right? Calories, fat, like, all right, this is the basics, but like, what about the non-nutrients? Non-nutrients are the phytoestrogens, for example, that can mimic estrogen like in the body. What are they doing and how do they impact? And would we need to know that? For my talk today, and for my research in general, it's on sialic acid, which is a type of sugar. So it's not a true carbohydrate, but it is a different type of molecule that has all of these different modifications. The complexity is enormous, and there are several different types that are across species. Humans have their own specific one. Other organisms have their own specific sialic acids. They are very important recognition for the immune system. They recognize self. I'll show you a picture to demonstrate the complexity in just a second, but they're very enriched in certain foods over more some than others. So I will explain U5AC, U5GC in a minute. Actually, that minute is right now. I can't. There's a picture here because I couldn't decide if I wanted to do the picture of the molecule first. But basically, we have this sugar, sialic acid. It's called N-acetyl neurominic acid. The neurominic part is there because it was originally found in cows and cow brains, actually. So, so the associated neuraminidase in the brain. So that's how the name got there. But there is an enzyme that most mammals have that convert our human sugar into a non-human sugar because we don't have this enzyme. So this sialic acid is pretty much foreign to us. So in the nomenclature of sialic acids, the human sialic acid here is denoted as purple, magenta almost. And then the opposite, the other one, which is just a simple modification of an oxygen atom here at the very end. This pointer is not the best, but here, only an oxygen atom is being added. And this is known as N-glycyl neurominic acid. So it's just a literal one addition of an oxygen atom to this molecule. And it is not naturally expressed in humans, okay? Because we've lost the enzymatic function. So as a result, we have an accumulation of the precursor, U5AC, and it's all over every single tissue in the body. And this is the image that I really wanted to show because this is why I wanted to kind of emphasize that glycobiology finds you. You don't kind of seek it out because in all my studies in nutrition and public health and all these things, we didn't talk about the glycocalyx at all. And the glycocalyx is what surrounds every single individual cell in the body. And in fact, the mass in some of these cells of the glycocalyx is larger than all the contents inside the cell of itself, which tells me that there's immense intrinsic complexity out here that we don't really understand because we've been so focused on the life's code. What is the DNA? What is the translation RNA, the messenger RNA, microRNA? What are those doing? And protein production. But we kind of left the glycans to the side because they are very complicated. The arrangement and different attachments, they're all done by enzymes, which are produced from the genes themselves, but it happens kind of stochastically. It doesn't remain in static. And that is what I think is so interesting because this is a fundamental property of every single cell alive today. And if you knock out the genes to make these glycan structures, it's embryonic lethal. It just doesn't make it, doesn't survive. So it's incredibly important. So this large density of network exists on endothelial cells, which line the vascular system of the blood, and epithelial cells like the intestine and the colon. So again, we lost this enzyme about 2 million years ago. No one knows why. That's a whole other separate talk we could, pontificate for days about. And the enzyme was fundamentally altered. So there was a significant chunk that is just completely absent, and it was selected for. And as a result, we still produce the gene, but it's so dysfunctional, it doesn't work. It can't add that hydroxyl group onto the human sialic acid. So all of our living primate relatives, our closest living relatives, express both because they still have that enzyme. Again, just wanted to show you here. This is the big chunk that we lost. Gone, no longer functional. But we aren't the only ones, okay? It's not human specific. It happened a couple other times with reptiles, alligators, platypus, which I think is an interesting example, turtles. And they actually lost it way longer before we did. So we lost it about 2 million years ago, approximately. Other clades of organisms lost it around 500 million years ago. And so of snakes as well. And so just here in the tree of life, right, we just a section of the tree of life, you can see here, that the loss of Nu5GC is indicated in this pink fuchsia color. New World monkeys lost it as well. Some white-tailed deer. Again, these have their own mechanisms and own, reasons for why that happened, which are really unknown. Humans as well, reptiles like chickens about 100 million years ago. Interestingly, some bats had lost expression of Nu5GC. So, in respect to human-associated diseases, if you have a virus or a bacteria that's coded in Nu5AC, it looks like self. And if it looks like self, you're more likely to get infected. If it had Nu5GC, right, it may be, oh, that's not self, that's not me. What is that, right? That's a whole other conversation in and of itself too, but we won't get to today, which I think is also really fascinating. So in summary, there is no apparent alternative mammalian pathway for Nu5GC, the non-human sialic acid synthesis in our cells. Maybe in cancer, there's some alternative mechanisms being proposed that it could be potential to be generated, but the preponderance of evidence does not suggest that is the case right now. So if Nu5GC is detected in humans, where is it coming from? Could it be our diet? Could it be the food that we grew up eating? I'm from the Midwest. I grew up on a diet of red meat, potatoes, and corn. I didn't think twice about it. But the reality is, going back to the nutrition label, you'll never see this on a package of meat, but the highest amount of Nu5GC is in beef. So here we have measurements of bound and free, which you can measure via when you treat your samples with heat, or if you keep them chilled to keep the bound and free. But I just wanted to show that this is, it's a very complicated issue, but in general, most red meat organisms that are consumed in the human diet contain Nu5GC, but there is some differential expression within this gene too. Certain types of fish, for example, salmon, appear to have this regulated only in the initial part of their life. So caviar is loaded with Nu5GC, but then adult fish, pretty absent. Very interesting. And as you notice, there are no known amounts, well, plants and veggies don't make sialic acids, but I just wanted to illustrate that there are no Nu5GC in plants or vegetables, or fruits or veggies. So back to this conversation of disease in America and associations. So again, we focused on colorectal cancer in the lab that I'm a part of, and associated labs look at atherosclerosis, so colon cancer, heart disease. But it just so happens that Nu5GC seems to be particularly accumulating in the endothelial cells, so the blood vessel cells, or epithelial cells, cells that line the GI tract. Unclear why that preferential selective uptake happens in those cells, but it's very interesting that the amount of food that we consume, the red meat that we consume, may be selectively pushing the sialic acid into tissues that make us more susceptible to certain diseases. There's a whole mechanism to explain why this is. For time today, I don't have time to go into it, but I think it's very interesting that in cancer, which is particularly known as a hypoxic state, so less oxygen in the state, less oxygen in the cell, when that happens, the sialic acid lysosomal transporter is upregulated, which seems to be driving more Nu5GC accumulation in these tissues. So to sum up again, when we consume a diet, with a ton of red meat, you're more likely to express Nu5GC on these tissues of cells. So you have this floaty, you have this connected sialic non-human sugar on your cell surface, and your immune system recognizes it as not self, and it mounts an antibody response. All humans have varying levels of circulating IgM, IgA, IgG antibodies to glycoproteins that contain Nu5GC. So, and it appears during the first year of life. So we are all exposed to Nu5GC at some point. Even maybe if you are completely vegetarian, right? If you come across animal dander, if you're on a farm, you could still be getting some exposure, which indicates that, maybe there's a balance here of having the right amount of Nu5GC antibodies to neutralize pathogens, right? But if we're chronically expressing it and making antibodies directed towards tissues, it can create a long-term inflammation or a low-grade inflammatory process that is important to be aware of. So this led to the general theory in my lab by G. Farkey, who created this idea of xenosialitis. So foreign sialitis, so clean sugar itis. Any itis you see is associated with inflammation. So that seems to be at the seated table of the interface of these antibodies that we are directing towards certain tissues and the incorporation of this non-human sialic acid. And could it potentially be associated with these various conditions that we're trying to combat in the US, but not effectively doing so? So with respect to finding this association and the known levels of antibodies, right, that we can detect, it would make sense that we would have a specific antibody associated with a specific disease. But that's not the case, because again, if you think back to the glycocalyx, everything that surrounds the cell, the variety that you have on the outside, you're going to make an antibody to this pocket, or to this pocket, or to this pocket. They all look a little bit different. And because they look so different, everyone kind of has a different antibody profile expression, and no specific epitope correlated with heart disease risk, which was, I think, an initial blow. It's like, oh, like we did all this work, and there should be an association here, and there isn't. So the thought was, well, the association between inflammation and 5GC is limited. But if you tease apart some of these studies, specifically the European Perspective Investigation of Cancer Nutrition, EPIC study, there were 71 colorectal cancer cases. And there was a positive association seen with colorectal cancer risk, but only when all of the antibodies were added together. So not just this one specific pocket or this one, the total. So the more antibodies you have against these new 5GC directed glycans, you're more likely. So if you're in a higher quartile, you're more likely to have colorectal cancer compared to those who had the lowest antibodies, right? And that in and of itself is a whole other discussion of why certain people express more than others, especially if they're consuming the same diet. Could it be regulated through the microbiome? Inquiring minds want to know. But again, we would need larger confirmation, right? Or at the very least, a different approach or a biomarker to establish this long-held association for years. And this here on the right is just an example of all of the different types of antibody responses that contain new 5GC. You can see there are myriad, and they vary from person to person to person. So the complexity here is just absolutely remarkable, and which is why we need a validated biomarker to help discern, where is the clear risk here? What is going on? And so, with respect to my project here, again, I had 5 minutes to describe this whole thing, and I know I just lost all of you at interfaces. So I'm really glad to have the time to go through this. But this is what I was trying to say at the last minute of my talk, which is what I'm focusing in on is chondroitin sulfate, which is a glycosaminoglycan, which is a long fancy word for just saying a repeating disaccharide. So 2 alternating sugars that go on and on and on and on and on. And this has been, well, for starters, it's made up of glucuronic acid and N-acetylglucosamine. So these are the 2 alternating sugars, and the modification here happens at the end position of this glycosaminoglycan. So this here is a glycol group, but normally it would be, if it is modified at all, it would be an acetyl group, because remember, nu5AC has an acetyl group. The human sugar, nu5AC, has an acetyl group. So the acetyl modifications would exist normally. Any modification that isn't nu5AC derived, for example, nu5GC, if there's a glycol group, it's going to be a little bit more foreign looking, like, I don't remember seeing that before. So, yeah, here we go. Here's the glycol group that I just wanted to show. The other one was the acetyl group. So this metabolism of nu5GC is thought to flow in this direction. And for sake of time, I'm just going to say that there's a preferential push of the non-human sialic acid from the diet down into this chondroitin sulfate molecule. So you can see here the nu5GC group, the glycol group in red, follows down these same pathways, which are the exact same enzymes that nu5AC is acted upon in this instance. It seems to be like an evolutionary holdover So it's, even though that hydroxyl group is still there, these enzymes are able to work and still cleave it and change it just in the same way. Again, because we lost this only 2 million years ago, so maybe evolution hasn't had the time to, create that, or the enzymes aren't as specific in binding, but it still happens. And there's this push towards chondroitin sulfate, where the glycol group from Nu5GC is incorporated onto one of these sugars. And no one knows why that push is. Like, why follow all of these paths? Why not stop sooner up the chain? Why not stop at hyaluronic acid? So it seems that maybe there is some use of chondroitin sulfate. And again, if you are a little confused about chondroitin sulfate, it's because it can be detected in the serum. We think about chondroitin sulfate in the bones and extracellular matrix of providing rigidity and structure, and but the turnover process allows certain molecules of chondroitin sulfate to be detected in the serum. Okay, so it's yes, it's in the bone, but it's also in the serum as well. And what have we found? Well, if you look here, you can actually detect chondroitin sulfate in all of these mammalians that we test. And so if you take the enzyme that makes Nu5GC and you knock it out, so it can't make Nu5GC, those organisms here, the cam null mouse, there's no detectable modified glycol groups on this chondroitin sulfate because the enzyme has been knocked out. We don't have the enzyme either, so it makes sense that humans wouldn't have it either. But some humans have trace levels, and because of that, and we don't have information on their diet, it's presumed that the Nu5GC that's coming from the diet and is metabolized is added onto this chondroitin sulfate molecule. So here it is demonstrating that. Oh, and then I wanted to point out shark too. Sharks also lost the ability to make Nu5GC, and as a result, they also don't express this modified form of chondroitin sulfate in their serum either. From a commercial chondroitin sulfate. So this is evidence of like, okay, clearly there may be a biomarker here that we can look at, but because the levels are so low, it's really hard to quantify with the technology that we have, even here at UCSD. Okay, we really need a very specific quantification system. So again, okay, so my project is in 2 sections essentially. We really want to understand if we're making antibodies against this compound, right? Because if we already make antibodies against a foreign sugar that's incorporated onto human surfaces, cellular surfaces, then it's very likely that we might produce antibodies against a modified chondroitin sulfate that wouldn't otherwise naturally exist in the human body. So the first part is to look for antibodies against this modified chondroitin sulfate that has this glycol group added. I didn't mention this either, but the GCCN here is the non-sulfated form of chondroitin sulfate. So chondroitin sulfate is, again, this disaccharide repeating sugars. Some of it is modified with sulfate. Others of it has no sulfate on it. It just depends on the section that has been clipped. And they range in variance in their length in the serum. So do we have antibodies against these glycolated compounds in the blood? And what does that mean for inflammation? Is it low grade? Are we still making an immune response, right? We're looking for immune activation because, again, inflammation is a driver for so many chronic diseases. It's really nice to establish the immune connection in these specific diseases. So that's the first aim in general. And then we can use either mice or chickens. And I think we're actually probably going to be immunizing chickens this year because it's not that mice don't make a bad model. When you knock out CMH and then feed them a Nu5GC free diet, they're a great model. But chickens seem to have more of an immune response because they lost Nu5GC over 100 million years ago. So that loss has really allowed them to have a more robust response. Certain mice that are cam-null aren't as robust as other types. So chickens make a good model, but mice also make an equal model. So depending on how you immunize them with the GC standard, which comes from an engineered bacteria, an E. coli that can produce this disaccharide, and then through chemical means, that's when the glycol group is added. That is being done at RPI, which is one of our collaborators, who have the more sensitive mass spec technology, which are going to be able to run the samples again, because we're picking up trace amounts, we really want to be with these small samples that we have, we really have to be very sophisticated in our mechanisms in which we're acquiring information, because these studies that we're trying to assess are built upon years of information. So the Nurses Health Study has 30 years of serum samples, but we only have like 150 to 250 microliters, which is a very small amount. And so for up until this point, most of the work has been done using like 500 micrograms or a milligram. So you have like enough to get the detection that you want, because these levels are so small. Okay. So that is one of the biggest challenges there, which is why we're partnering with RPI. But again, we want to know if we can make these, we want to know if we can make antibodies against these in the mouse model to see if the immune component is involved here. And we can repeat this and then get monoclonal antibodies for the generation to do secondary detection, which is another partner we would use. And then to check, obviously, to make sure that our chickens don't have Camnol, but mice do. So to confirm that, you can do a Western blot and you can do tissue immune staining chemistry, take a little sample, section it, stain it with lectins and see if it has some 5GC in it. You can also digest with chondroitin for another control. For time, I really want to talk about, 1 second here. Tissues. Okay, same thing. We can get samples from tissues across various labs, which do have noted cancer, and stain them to detect in the tissue. Again, the point though is for more of a biomarker, like a readily accessible, non-invasive biomarker, which is obviously serum. Tissue samples, that is pretty much as invasive as you can get, but it's something we can also look at, and it's interesting because we don't really know what chondroitin sulfate looks like in the extracellular matrix, what it's doing out there. So it could be very valuable information. And again, for the same thing for the mice, you can use paraffin or frozen sections, slice them, stain them to detect where it is, if it's present at all. And then I wanted to talk about this part. So the first part, antibodies, immune activation, immune system. Second part is, okay, now you have all of this data from various studies, long-term studies to assess follow-up of red meat consumption for over 30 years. What do you do then? So from the study population, we use the Nurses Health Study, which is 1 and 2 are just 2 different arms of it, but mostly nurses who have very good follow-up. And then this one, we needed male providers as well to get an equal sex ratio here. So those are our healthy samples, but within those, we have patients who have disease, not just colorectal cancer, remember. So for my minor proposition, I focused on colorectal cancer, but we have so much information on these individuals. We may find that there could be another association, right? It could be stronger with heart disease, maybe rheumatoid arthritis. There's a link there with red meat consumption and rheumatoid arthritis. So we can start to tease apart, but just for what I presented, it specifically focusing on colorectal cancer and heart disease, because those appear to be where there's the preferential push of the new 5GC into those specific cell types. Again, lots of information here, excellent follow-up, 94%. That's everyone. I don't know if any of you have done clinical trials, but like to get 94% follow-up is so challenging. And death follow-up is 98% complete. So these are probably some of the best samples you can get for information. That being said, I mean, they do only update. It's a dietary pattern assessment. So that's done qualitatively every 2 to 4 years. So it's just generally capturing the scope of the diet, but not like fine-tuned, right? Because we know nutritional patterns change over the course of the month, over the course of the season. If you're lucky to live in California, you can eat seasonally. But in general, they're filled out pretty consistently and accurately. And these studies are well powered. And within these studies of that sample, we have the combined Nurses Health Study. We have about 500 women who went on to develop colorectal cancer, and we have about 400 or 350 men from the other study who developed that. We can actually take these serum samples, process them, treat them with chondroitin sulfate, isolate them, and then pack them up and ship them to our collaborator, RPI, who has that more sensitive mass spec technology to detect chondroitin sulfate, which they're also making them standard. So when they run it, they'll run the standard, and they'll run chondroitin, they'll run chondroitin sulfate, and they'll run it with the modification and without the modification to see what it looks like in these populations of people who develop disease compared to those who didn't. And we can track this over time, which I think is really, really interesting. Again, in conclusion here, there's a lot of considerations for this, which is why the statistical analysis component is from it's going to be added to our support from Harvard, which is how I found about Ajit Varki and glycobiology in general, because they're very interested in looking at this. So they're going to run a lot of statistical analysis as well, some numbers now. And yeah, they can do stratified analysis as well. So further break it down into subsets and do the proper, approach for that. And again, as I said, these are well powered because we have such a large sample size over long term that can allow us to really see what could be going on. And I think that this is very exciting because, as I mentioned in the beginning, we don't have any biomarker of risk yet for any dietary patterns, just suggestions on you should do this, you should do that. But diets are incredibly nuanced and not everyone has to eat the same things. Other people run better on, high fiber diets, where some people are extremely more sensitive to even increasing the fiber content. So the clinical approach in guiding nutritional patterns to optimize health and to prevent disease, right? That's why we're all here, trying to prevent things from getting worse. This can start to guide that or lay the foundation for approaches to figure out, okay, let's maybe see how we can intervene here, especially if you've already developed colorectal cancer and are at risk of developing it again by following similar patterns. It's nice to know where you are, where you fall on that line, and assess risk from there. You can integrate this information with metabolomics, yes, but glycol analytics I think is going to be huge in the future because there's just so much information in the glycocalyx that I think it's going to be very critical in the future. Because again, these things have their unique signature across cell tissues and body types and between sexes. So lots to discuss there. And in the end, I want to leave you that the integration of this new data will help us gain insight into, the origins of disease and strengthen the biological possibility of this concept of xenocellulitis and its many manifesting forms that we may not know yet. And yeah, if we can find a biomarker for colorectal cancer risk and start to implement it, I will be beyond excited. And that is why I'm here at UCSD. So thank you so much. Happy to take questions. I had a lot of questions when I first heard it.
I actually was going to ask about rheumatoid arthritis before you mention it, because cartilage has got a lot of chondroitin sulfate in it. Rheumatoid arthritis is an autoimmune disease, and it's characterized by acute inflammation. So it could be a good target. But I had a different question also, which was, are there any groups of patients that you know of, or that you might discover by screening the patients who have high antibody titers, that seem to have acute sort of inflammatory responses to red meat? Because if they were, particularly if they had, say, rheumatoid arthritis, then what you could do is you could engineer a transgenic pig that didn't express a new 5GC and then see if they still got that response.
Yeah, I don't, any of you have heard of people who, like, anytime I travel outside of the US, if I eat, dairy or bread, if I eat it in another country, I'm fine. I come here, I'm wrecked. It could be that. I do think that we even know that there is variability among different cow species. So I showed that new 5GC content is highest in beef, but that's American beef. There are I think there's a type of Mongolian cow which has very, very little new 5GC. So when you consume it, you would have very little expression in tissues. And so that I think is really fascinating. So I think for that information, though, we would have to have information a part of the dietary studies that
Yes.
suggest, like, I follow this diet because I'm sensitive to,
Yes.
red meat or beans. But you lose that with these very strict questionnaires. Again, for nutrition studies, they are only as good as the input that has been given. And my first rotation at UCSD, I was studying oxalates. Oxalates are this little compound in all types of fruits and veggies. And there's certain bacteria that can break it up. And that's great. But if you don't have that bacteria, you can have these oxalates accumulate in the blood, which can increase your risk for stones, right? Terrible, terrible stones. So I was looking at this dietary assessment on oxalates, and it was the worst. It was the worst assessment I've ever. Every group had oxalates in them, and they weren't even separated out. So when we put in all the data and crunched it, there was a little trend, but like, for example, like chili was one of the food. Chili, beans contain a ton of oxalates, but red meat doesn't, but they're mixed together. And so we can only extract so much information from well-designed nutritional questionnaires, which these questionnaires were made 30 years ago. So yeah, it is a severe limitation. But interesting nonetheless, because I think we all eat food and we all know what reacts with us and what doesn't. But there's mechanistic explanations for why that is.
Well, let's thank Will and Justin. And thanks, Kelly Malone.
I made sure to provide vegetarian sandwiches today.
Thank you. You
Thank
can
you.
still eat fish and chicken. There's no meat in most of those.
Okay, thank you, Dr. McCullough. Thank you, those of you who joined on, those of you who joined us on Zoom. Thanks, everybody, for coming. Make sure